Hello biology lovers, welcome back to Cell to System. In today's learning, we will be diving into the most fascinating topic of plant biology, that is fertilization in flowering plants. So whether you are preparing for your board examination or any competitive examination, this is a must know topic in your syllabus. So without wasting any time, grab your notebook and let's begin with our today's topic. What is fertilization? The simple definition of fertilization could be the fusion of both gametes, both male and female gametes. In case of flowering plants, the male gamete is very small and motile and the female gamete is large and non-motile. This kind of fusion is known as syngamy. So we will be learning syngamy. Before that, we have to know some other preparatory phases. If any compatible pollen grain falls on the stigma, it has to go through various phases. Let's begin with those phases. First, what will occur? Male gamete. The first thing which will occur is the formation of male gamete. When we have studied about male gametophyte formation or microsporogenesis, we have came up to a specific phase, that is mature microspore. This mature microspore grows through a mitotic division and forms, this is N, and forms one small generative cell and another large vegetative cell. This generative cell, this again divides to form two male gametes. We will solving the question that is in your mind, that why the need of two male gametes here. So we will be answering this question later on. Now this large vegetative cell will be forming the pollen tube. Some plants, when the pollination occurs, up to this phase is done, but in some cases pollination mostly occur in mature microspore stage, where we are taking the condition that pollen grains have came upon the stigma, have fall upon the stigma. Pollen grains, they have a sculptured exine, that's why I'm drawing it like that. These pollen grains have fall on the stigma. Next phase is germination of pollen grain. When the pollen grain, it falls on the stigma, it germinates by absorbing some sugary stigmatic fluid from the stigma and it becomes swollen. This swollen pollen grain will be forming the pollen tube. So if this is the, if here is the germ pore, this pollen grain forming grains, the pollen grains, it becomes swollen by absorbing stigmatic fluid. These are some sugary fluid. In the next phase, pollen tube is formed from, remember germ pore? The germ pore was present in the pollen grain. The discontinuity in the sculpturing of pollen grain is the germ pore. This pollen tube, it penetrates through digesting the tissue of tile. Next phrase is entry of pollen tube into the ovule. Entry of pollen tube into the ovule. So what really happens? Let's understand here. So this pollen tube was forming. Here it will, this is the micropyle region. It will come to the micropyle and go through the micropyle, and it will go into the enter the ovule. This is the ovule, integuments, micropyle. This is the ovule. Type in which there is in fact three types in which way the pollen tube can enter the ovule. I have already discussed about the three types, but again I am saying one is porogamy. Porogamy means entry of pollen tube through the micropyle. Entry through the micropyle. Next is telazogamy. The entry through the chalazal region, it will come like that. It will just come like that. It will come here and go in this way. But in our study, we will be learning about porogamy. We are considering the case porogamy through the chalaza. And in case of mesogamy, there is another case, mesogamy, in which the pollen tube will enter the ovule through integuments. Up to this, these are the preparatory phases. But now I am going to start with the main magical phase, which is very unique to angiosperms, that is double fertilization. So let's begin with that main part. This double fertilization process was discovered by Naustin in 1898, and it was also supported by Strasburger and other scientists. So here we need to understand why the term double fertilization. In this phase, the fertilization occurs twice. We will know how. First, what happened? The ovule is penetrated by the pollen tube. What the ovule really has in this phase? It has eight nucleated embryo sac. These 3 cells at the micropyle, 3 cells at the chalaza, 2 nuclei at the center. Now, let's understand what they are really called. These 3 cells at the micropyle, they are known as egg apparatus. These three cells at the antipodal region, they are known as antipodal cells. And these two nuclei, these are known as Pollen nuclei or secondary nuclei or definitive nuclei. Let's understand their function. Here, this egg apparatus, it contains two cells and one egg. These cells, they are known as synergids. As you can see in the diagram, these synergids, these two are synergids. I have drawn with green color to differentiate them from the egg cell. They are synergids and egg is somewhat different from them. These synergids, they help the pollen tube to enter through the ovule and fertilize the egg. Their function is entry of pollen tube. These three cells, they are alike and they are known as antipodal cells. Their function in some books, it is written that the antipodal cells help the pollen tube to enter through the chalazal region in case of chalazogamy, but in some books the antipodal cells doesn't have any function or the function is unknown. Now pollen nuclei, they are responsible for the formation of endosperm. When this fertilization process occurs, when the pollen tube enters the embryo sac, one male gamete fuses with this egg. For example, this has one male gamete, this is the male gamete, this fuses with the egg and it forms diploid zygote. Another male gamete, as we know the generative cell has two male gametes, another male gamete fuses with two polar nuclei forming triploid. As we know that if there is fusion of two nuclei, it forms diploid, and if there is a fusion of three nuclei, it forms triploid. Triploid endosperm. And what is the function of endosperm? Endosperm is the nourishing tissue for developing zygote or developing embryo. Two consecutive fertilization occurs. Here I can also draw. So this is one fertilization and this is another set of fertilization. Two fertilization occurs. That's why this process is known as double fertilization. This process is known as double fertilization. So let me know in the comment section who is the discoverer of this process. I have already told in the video. So if you have watched the whole video, you may know. Let's have a quick flow chart or quick revision about the fertilization process in angiosperms. What is happening? The pollen grain and stigma, they are coming in contact. Pollen grain is forming two male gametes this one male gamete, this fuses with the egg cell forming zygote, which is diploid. This another male gamete. This is fusing with the polar nuclei, forming primary endosperm nucleus. First, PEN is formed, then primary endosperm cell is formed, and then finally the endosperm is formed. This endosperm is the nourishing tissue required for the development of a embryo. This is all about the fertilization process in angiosperm. In the next class, we will be starting with the next topic, which is post-fertilization changes in the angiosperm. So, if you want to continue with the topic, you have to stay tuned with Cells to System. Stay safe, stay healthy. Thank you.